Good afternoon, everyone. My talk today is talking about a case study where we used a custom T-cell that Promega made, and it works quite nicely with Jamie's talk earlier. So before I go into the case study, I just wanted to give a quick introduction into my company, Reading Scientific Services, or as we often call it, RSSL. And then I'm aware that not everyone works to the GMP regulations, so I just wanted to give an introduction into the analytical procedure lifecycle that we use within a GMP space. I'll go into the case study with some data that my clients allowed us to share, and then talk a bit more about the early phase qualification that these cells went into. So RSSL, we're a analytical CRO based in Reading, as the name would suggest. We're actually owned by Mondelez International, which is a $25 billion food company that makes well-known brands such as Oreo and Cadbury's. The advantage for us to do that is that we have a lot of investment into our equipment and into our scientists. Historically, we were servicing the food science sector with contract research, but now about 80% of our work comes from the life science sector. And most of those clients come from predominantly the UK and Ireland, other European countries as well. But we also do work with USA and countries from around the world, generally on their the pre-GMP, the development type work. As I touched on, we cover all phases of the drug process. So we do characterizations for GMP batches. We also could do impurity isolation and identification. We help with preclinical development. So currently we have some formulation studies ongoing within my department, but we'll also do force degradation, method development, and things like that. Predominantly because we are GMP accredited, we do a lot of method validations and GMP compliant stability studies, and we'll also assess extractables and leachables. And then we have quite a large couple of departments that do compendial testing to all the pharmacopoeias. So we're set up to help with any analytics that a company may need. And just this is a really, it's a top level summary of all the various methods that we cover at some point within there. I've highlighted in bold the methods that we would routinely use to characterize a drug that would predominantly be used for a bioassay. So usually it would be a biopharmaceutical. So we'll employ the biochemistry techniques. We'll also do some separation science, such as peptide mapping or intact mass, or post-translational modifications, just to ensure that everything is working as it should. So moving on to our the analytical procedure life cycle. The ICH, so that's the International Committee of Harmonization, have many documents that tell you how to ensure that your product is how the manufacturing is controlled, how the product is assessed for quality and things like that. There are currently 2 documents which they're in draft form at the moment, but they are in the process of being ratified, so they will become a legal requirement to follow in the future. And these 2 documents are the Q14 and the Q2, and they're designed. These 2 documents are designed to work together. So the Q14 is a brand new document, and that will it describes how you manage your method throughout the process and how you control that the method is performing as you would expect. And then the Q2, this document's existed since, I think, 1995. It's quite an old document, so they've written a brand new version of it, which is designed to be worked in conjunction with the Q14. And then the Q14, it describes the analytical procedure life cycle in a lot more detail, but I've just taken one of the figures which shows the life cycle in a nice visual format. So to start with, this is the process and understanding, and then the control strategy. So this is the scoping stage of deciding what sort of analytical method you might need to employ for your drug substance. You then would have to write an analytical target profile, so that would just tell you your precision parameters or your accuracy, your linearity, what working range you want, and various requirements that you would want to develop your method to be able to complete. And then that also defines whether you would choose to do a ligand binding assay or a mode of action, for example, T-cell engager type assay. And then you go into your development phase. Quite often with this new Q14 document, you actually do a risk assessment. Last week I spent a 5-hour meeting doing an ELISA risk assessment. They're quite laborious management documents, but I suppose in terms of a regulation point of view, it means that auditors would have everything that goes on in a scientist's head actually on paper. And then you undergo the normal development process. Then we would go to this validation stage is where the ICHQ2 document would be applied. And then once a method is validated, it can be used for the relevant stage that you validated the method to. So for example, phase 1, phase 2, or phase 3. Or if it was going to go to market, if it was going to be commercialized, then it would be the final release testing. But like everything, you have to monitor it continuously, and occasionally you need to change something or improve the method somehow. For example, if a key reagent's changed, or in the case of cells, a slight change in a working cell bank might mean that it needs to be revalidated. In which case you go back to make your working cell bank, and then you come back down to the validation. So when you hear the analytical procedure life cycle, this is the whole concept that they mean. That is just this continuous cycle of development, validation, and then change management if it's required. For those of you who are more familiar with the GMP regulations, my use in the title of early phase qualification could be quite controversial, simply because in the documents they regard qualification as being it's the qualification of a manufacturing procedure or a software or some process involved in the manufacturing of the drug rather than the method itself. So I just wanted a slide to demonstrate what I mean in this talk as a qualification. So this is sorry. So this early phase, the qualifications in terms of the procedure or the method is normally done at the early phases. So for example, we quite often do it if we're working with a preclinical substance and we're developing a method that will be used for a final release test. So it might be a stability indicating method, or it might be a toxicology batch that they want us to develop the method before they have the GMP batch to validate it on for phase 1. We do sometimes do it for before a validation as well. In that instance, we would sometimes term it as a pre-validation check, and it's just essentially a method qualification would just be to make sure that your method design is working, and it tells you whether you need to optimize it more to get better accuracy or better dynamic range or better sensitivity. The validation itself, this is defined by the ICHQ2 document. So this is mandatory for the later stages of drug development. So to get something into phase 2, you would have to validate all your methods. And depending, we call it phase appropriate validation, because depending on how far through your drug life cycle your candidate is, it depends on how much work you have to put into the validation to prove the robustness and everything is required. And I suppose more importantly, you don't actually have to do any pre-validation or any qualification of the method before a formal validation. So now I'll move on to my case study. And as part of this, I've just thought I'd show the life cycle diagram to just indicate which parts of the process we're going through at the various stages of this case study. So the brief, this is the initial scoping calls with the client, and in this case it would be the product and process understanding. As we're a contract research organisation, we don't know the drug intimately until we've had enough discussions with the client as to what is needed. So in this case study, the client's technology is a therapeutic vaccine that's designed to be processed by the cells that results in an immune response. Essentially, it will get presented to the immune system as you would expect. And one of the drug candidates in particular was designed against to be a vaccine against the human papillomavirus. So as part of the scope, we were tasked to develop a mode of action T-cell activity assay to demonstrate that the vaccine could actually activate the T-cell with a specific receptor to this E7 11 to 19 epitope. The other specific requirements that the client had was that the custom T-cell would need an IL-2 luciferase reporter gene to actually show activation rather than binding, and that an antigen-presenting cell should possess dendritic cell qualities. So sometimes within our lab, the concept of the target and the effector cell is not always known. So I just wanted to, before I go into all the detail of the method and everything else, I just wanted to give a couple of definitions. So the target cell line, by that I mean it's the antigen-presenting cell. It is targeted by the therapeutic. And then the effector cell line, nowadays it normally contains a reporter gene. Historically, it might have been a radioactive type method or something like that. And it produces the measurable function. So generally with these activity assays, it's the luminescence. You do have, obviously you still have killing assays. So the talk before was showing about the killing T-cell method. That's still functional, but when you're release testing a drug substance or a drug product, sometimes you need it to be, I suppose you need to be able to control it in a lot of detail, so it's less used in terms of a GMP release test. And then HPV, I'm sure most of you are aware of HPV, but again, I'd just like to give the definitions before I go into detail. So it's a high-risk strain, and it causes expression by the E6 and E7 oncogenes. So the E7 peptide, which the client wanted to target, is the immunodominant antigen, and it's expressed by this MHC allele, the A0201 on the cell surface. And it was sequenced in this paper as well. So as part of this product and process understanding stage, we had to source the T-cell, and by that we thought of different options, whether doing it in-house or whether to try and outsource it and get a different supplier to do this. And the reasons why we chose Promega were several. So we have prior experience with Promega bioassay kits. So this graph here is showing a T-cell engager assay with, so the red lines are reference standard, and then there's two forms of aggregation that are impacting the potency of the assay. And this T-cell effector assay is using a NFAT JERCAT T-cell kit. And we've been using this for a GMP assay for probably about 4 years. So we knew the NFAT JERCATs were working very well. We also had, as this client had approached us, we had just validated a co-stimulation assay. So in this feasibility graph here, essentially it's a CD3, CD28 co-stimulation molecule, and we started using the IL-2 kit with the same from the same T-cell activation product line. Around the same time of this project coming in, we also became aware of the knockout cells that Jamie was talking about a bit earlier on. And so the fact that the T cell receptor was knocked out and that allowed you to do a transfection or transduction of your chosen T cell receptor made it a very promising solution to this method. And then in discussions with Promega, it turned out that at that time the E7 T cell line was available to purchase. So I've just put up here some of the R&D data that Jamie had shared with me, showing that the CD8 and the specific T cell here is in the population, so it's quite a clean population. And also, I suppose from a GMP point of view, the passage stability was very good. So you can see here between passages 9 and 21, you're getting the same curve shape. And a GMP is all about control and reliable, robust results. So that made it a very interesting selling point for us. Now, Jamie's talked a lot about this. We found that a lot of work had to go into differentiating the antigen-presenting cell, probably in the same fashion as the Promega lab. It probably took the whole project was about a year long, and developing this differentiated APC was probably about 4 to 5 months of that year. It was a lot of work. So to avoid issues in a final GMP method, we had several target criteria for the antigen presenting cell. So firstly, we wanted to use an immortal cell line, mainly because they're more consistent in their when you passage them, you understand them a lot more than a peripheral blood, and it just allows for adding more control measures to ensure quality of that cell line. We also, during development, tried various monocytic and dendritic-like cells and cell lines. Many of these needed differentiating before use. Unfortunately, because we're a contract research organisation, my client, although they're happy for me to share the data that we've received, I'm not allowed to share the cell lines we've used or the differentiation protocols, just because of the confidentiality reasons. Within RSSL, a client's method is, a method developed for a client is the client's property, not ours, so we had no control over that. Despite that fact, I've put in just for the 4 optimal differentiation protocols that we tried, I've put in the results that we obtained with the E7 T cell line, just to show the differences that various cytokine protocols can make. And all these protocols are available in the literature. They're just we just can't tell you what final paper we used. So as you can see, we tried the 4 different differentiation protocols, and then we also tried a 6-hour incubation with the T cell, and then a overnight incubation with the T cell. And as the drug substance would require presentation in this assay, we were looking for upregulation of the presentation of the peptide, so we just used our E7 peptide control. And you can see that differentiation protocol 4 has quite a remarked increase in signal compared to differentiation protocols 1 and 2. And so protocol 4 is what we moved forward to the final method, which sort of shows here. So for protocol 4, we would add the mixture of cytokines, and then we'd allow them to differentiate for about 3 to 4 days. We'd then remove the spent media. Again, because it's the client method, in this diagram I've just put the positive control as that's used to just make sure that the T cells are binding correctly. You then add the T cell effectors, and then measure the luminescence once you've added the substrate. For this project, we decided to go with an analytical method qualification, predominantly because this was a early phase proof of concept method. So we didn't actually have any GMP-ready drug substance. So it made more sense to do a qualification rather than try and do a phase 1 appropriate validation. Before I go into the data, I don't know if many of you have seen the ICH Q2 guidelines, but essentially in both the old version and this newer version that's coming out, there's a table that defines what criteria you should assess for a validation or a qualification. So for an identity method, so this is just a yes, no, is your protein present? You would just assess specificity to prove that it's detecting the right thing. If you're looking for an impurity, you have to assess it as a quantitative method. And I suppose more importantly, your limit of quantitation and limit of detections are quite important for impurities. And then this method comes under the assay content and potency. And so again, this is a full quantitative validation. So we look at specificity, linearity, which falls under working range, accuracy, precision of the method. So here I've just taken a dose response curve that was taken from one of the validation runs, and this is just using the E7 positive control. So it's just the 9 amino acid peptide, and it has the finely differentiated antigen presenting cells. This one was the positive control was added, and then the T cells are added. So you can see quite clearly that there's a nice dose response curve, a good 4-parameter curve fit, and a decent range between the lower and upper asymptotes. For specificity, this is the data for the specificity of the Jurkat cells. So to measure that, we used the E7 peptide, and then we created a scrambled E7 peptide. So essentially, we took the same 9 amino acids, and we just randomly changed them so that if any binding occurred, it was not because of the order of the amino acids. So you can see we get we get the good signal with the E7 peptide, and we get some signal, but not a great deal with the scrambled E7 peptide. And then our media-only control is showing no signal whatsoever. So it's showing that these T cells actually do have a good specificity to their target antigen. In terms of linearity, because I'm only allowed to share with you the data from essentially the positive control, I've used techniques we might use to assess linearity for ligand binding assays, because effectively what's happening with this 9 amino acid peptide control is that it's binding to the MHC molecule, and then the T cell receptor is binding. So it's a very complicated 3-step ligand binding, but it is effectively a ligand binding assay, as I'm showing you. So with linearity, we've just plotted the expected relative potencies. So generally within our GMP bioassays, we would say we fix the potency of the drug substance, say at 100 nanogram per mil, and then we would assess relative potencies by creating the reference drug substance at between 40 to 160 nanograms per mil, and that will show us that if a manufactured drug substance had a drop in potency, we'd still be able to detect it. And what you would expect is that at 40 nanograms per mil, you're still getting a measured potency of 40 nanograms per mil. As you can see, that for linearity-wise, our relative potencies are changing as we would expect, showing that the binding to this T cell is in a linear fashion. Again, in terms of accuracy, normally for a GMP T cell activation assay, we would measure actual potency against the reference standard of the drug substance, and then the drug substance batch that uses the validation. Because we couldn't do that, and it was just a qualification, we're just showing that the positive control, you're getting back what you're expecting to put in. So we've assessed the percentage recoveries for each of the different concentrations, and again, depending on which document you'd use, generally it's 80 to 120% is a good recovery for a ligand binding assay. So So you can see that it's binding very well to the E7 peptide. And finally, for those of you familiar with ELISA validations, this precision profile, again, because we're effectively showing its the ligand binding performance of the T cell, this precision profile is used quite a lot in the validations of ELISAs. On the right-hand axis, we have the percentage CV at each point, and then we then have the blue. This line here is the percentage recovery. So the precision profile shows you what the working range is of your assay. So essentially, as long as your percentage CV, so this is your intra-assay variation, and your recoveries are within your criteria, in which case, in this sense, CVs was less than 15%, I think, and then the recoveries was the 80 to 120, then that is shows your working range of your method. So you'll notice that this axis here, it drops. We had two other standard points closer to 0, but our recoveries were reading about 900%, which shows that actually that was within the lower asymptote, and we weren't differentiating that between noise. So in summary, we found the E7 T cell line to show very good specificity to the E7 peptide. And then when we added the E7 peptide at varying concentrations, we saw good linearity, percentage recovery, and precision. And also, it's not in the data that I've shared, but we've actually, for this qualification process, we use cell line passages between 13 and 20, so that correlates nicely with the R&D data that we originally saw about the stability of this T cell line. And then just to highlight other types of cell bioassay that we do within RSSL, I just thought I would just show a couple of feasibility slides from functional potency assays. So in this slide, we have been trialing the elite access interferon alpha reporter cell line. So we have a drug candidate that requires us to measure the amount of interferon alpha binding to the receptor. This reporter cell line has the reporter gene is the ISRE, so that's the promoter that's linked to the interferon alpha beta type binding. So this experiment was just showing we're just checking to see whether the overnight method was suitable for our purposes. So we added interferon alpha as the reference standard. We also added interferon gamma and TNF alpha just as negative controls, just to see how it's performing. And you can see, so this was an overnight incubation, and then we added the substrate the next morning. So you can see that there is a dose-dependent shift in the line. So although all of them are producing positive signals, there's about I think we worked it out to be about a 10-fold change in response between the interferon alpha, TNF alpha, and interferon gamma. We think it's due to the overnight incubation, that had we optimized it to be a 6-hour incubation, then we would see less interferon alpha or TNF alpha. And we just think it's because this cell line doesn't have any knockouts, it is just the inherent redundancies that cell signaling has, and there's a little bit of crosstalk. The other cell line is the NF-kappa B cell line, which we've been using just to in this instance, we were just using TNF alpha as a it's a classic. If you go to very old cytokine cell bioassay textbooks, the NF-kappa B is the signaling pathway that TNF alpha will activate. So we use that as the positive control, and as a negative control, we used IL-4. This time, to try and avoid any potential crosstalk from similar cytokines, we used one that uses a different signaling pathway, just to make sure that the internal signaling is functioning correctly. And as you can see, with here is a nice dose-resplendent curve with a TNF alpha, and then the IL-4 is a pretty much a flat line, which is very good for our purposes. So in summary, the new ICH Q14 and Q2 documents, although they're yet to be implemented, they're designed to be used together. And the idea of this whole life cycle is to ensure that the analytical method is a good, reliable method, and that it's continuously reviewed and monitored rather than you validate it once and then 10 years later there's a problem because you haven't been monitoring it. And also we found within RSSL that the Promega cell lines that we've used, they've become a good cornerstone of a GMP validated method. Some of these cell lines, they're in our clients' IND submissions and are used for releasing phase 1 and phase 2 type drug substances and drug product. The HPV T cells, the E7 T cell line, for a custom T cell line, it shows that it behaves very well under GMP conditions. We've both, within RSSL, we've used both the cell propagation models and Thorand use. And in some instances, we've tested, of different kits, we've tested Thorand use and cell propagation, and we find that the performance is very similar. And then I suppose this is just a watch out for anyone using cytokine binding cell line models, just in general, really, that there are so many redundancies that you may be susceptible to crosstalk, and so choose your controls carefully. And then I just wanted to thank my department, the Biomolecular Analysis Lab, and also in particular Matt, Pinaki and Laura, who did a lot of this work. I won't embarrass Matt, but he's in the audience. Any questions?
Thank you. Really nice overview. So you said that your client wanted to do also an IL-2 assay from your... I understand.
No, so they wanted to show T cell activations. They wanted to show that the T cell receptor was activating the T cell. So they wanted the T cell to have the IL-2 promoter, a fusion protein with luciferase.
Okay, okay. Because I was thinking, well, I misinterpreted that, or my head went on a different path, and I was thinking whether you would have been, well, now I'm doing the question anyway, so whether you would have been able to use the same reporter cell line to assess IL-2. So you would have been able to use the reporter, and also the cell line would still, I don't know if that's a question that you can answer, or maybe Promega, the cell line still produces IL-2.
I don't know. So I suppose I'm not a T cell signaling specialist, but from my understanding, because it's producing the luciferase, I don't think it would produce the IL-2. But I imagine Jamie or someone in Promega might be able to answer that better. Sorry to pass it back.
So yeah, the jerkets will produce IL-2 after they're activated, but the signaling pathway would be different than what Alistair is measuring. So you'd need a STAT5 reporter in that T cell to see that IL-2 is signaling. So he's really just measuring the T cell activation from the TCR, not the IL-2 production.
Thanks, Jamie.
Thanks. Hi, really nice talk, thank you. Just a question about the cell lines you're using. You said you used them between passage 13 and 20.
Yep.
Have you tried going above that? Do you notice any drop-offs as you get older?
We try not to. So in feasibility, we did look at it, and we didn't. The cells looked healthy above that, but generally within GMP, I think regulators would get a bit nervous if you're getting close to the senescence point. So we try and just use sort of the what is commonly accepted as reasonable passage numbers. So.
Great, thanks.
Does ICH-Q2 tell you how to validate or what to validate?
It tells you both. So that table that I showed you tells you what you should be validating, And then the new revision actually has some nice appendices that tells you for bioassays what should be in a bioassay validation. And then it also tells you how to validate. There's lots of text. If you've read the older one from the 90s, some of the text hasn't changed, but it clarifies it a bit more. It makes life a lot more easier if you've never had to validate before.
Okay, great. Thank you very much, Alistair.